My primary interests are in the interactions between Vibrio cholera and predatory bacteriophages. And I was studying this from a coevolutionary perspective as well as an interesting mechanistic kind of example of phage-host interactions that are important in human disease potentially. And at that point I discovered a phage that had a CRISPR-Cas system that was using it to target an anti-phage defense island in Vibrio cholera. So sort of exactly the way CRISPR-Cas is used in bacteria, but just in the opposite direction. So the phage is using it to target the host. And so I definitely didn't expect to discover that. It was very much an accident. It was one of those things where I had sequenced the genome, got to the Cas genes, did the blast, and thought, there's a mistake here. But that's how I sort of got into it. Yeah, I mean, for us, it was a really clear indication that the phage was interacting with Vibrio in a way that was probably saying there was constant antagonistic coevolution going on. So it was a really sort of amazing example of what phage would do to overcome bacterial defenses. And I think that this is important both from sort of a perspective of trying to say, for sure this is happening, not just because I co-isolate them, but because we can see now on a molecular level, there's evidence that the phage was being selected upon to the point where it had to have a CRISPR-Cas system. Says that those interactions are likely very important. And also from the perspective, not that myself I'm interested in phage therapy, but I think it's really important that we look to natural systems like the one that I study to look at how phages and bacteria are going to co-evolve, antagonize one another in a real world setting where there's constant exposure to mobile genetic elements. And so the kind of things that you expect for like resistance to build up and counter resistance to build up is going to be very different than in vitro, I would say.